Hello friends, today we will be discussing about the emerging technologies for cancer biomarker discovery. With the advanced knowledge on cancer genomics and proteomics, we can develop effective biomarkers which lead to the better understanding of the neoplastic development, its diagnosis and prognosis. The discovery of biomarkers improves the diagnosis and screening of the disease. It helped in getting a better insight into the personalized medications, real-time evaluation of the drug treatment progresses, measuring the genetic and proteomic profile changes in the patient before and after the treatments, and also it helps in identifying new drug targets. We'll now discuss about few techniques used for genomic and proteomic profiling that had facilitated cancer biomarker development. First of all, microarray, the technique which can identify thousands of different DNA samples based on nucleic acid hybridization techniques. Here on a glass slide, single-stranded DNA probes are attached to the uh, where each probe represents individual genes or the DNA spots. The cDNA synthesized from tumor cellular mRNA are allowed to bind to the respective DNA spots and detected by specific detectors which will generate quantitative signals that can be compared with a normal cellular microarray. This technique is very useful in detecting quantitative gene expressions between normal and cancerous tissues. Next is the SAGE or serial analysis of gene expression, which is a quantitative analysis of large number of transcripts. Before the SAGE, RNA hybridization or expressed sequence tagging were the methods for studying gene expressions, but those are very time-taking and limited in their uses. SAGE enables sequencing large number of transcripts at the same time. Here, after isolating the cellular mRNA, they reverse transcribed and processed with a series of restriction enzymes and specific primers with tags. Now, after the processing of each of the mRNA, they will be having specifically tagged nucleic acid sequences which can be joined together to form a concatenaric sequences, which can be cloned and then can be sequenced with the help of simple sequencing technique like the Sanger sequencing. The sequencing can give us the quantitative details of the individual gene expressions owing to the presence of specific tags. Hence, a single sequencing can generate data about thousands of gene expressions. While both microarray and SAGE are utilized for detecting gene expressions, there are certain differences, advantages, disadvantages in them. SAGE sampling is based on sequencing mRNA and not on the hybridization of mRNA to probes like microarray. Microarray is much cheaper, whereas SAGE is much more accurate and error-free. A variation of microarray is microRNA microarray or the miRNA microarray, where the cellular microRNA pools can be analyzed. We know that the microRNAs, they play a huge role in gene regulation, hence it has got immense importance in cancer diagnosis. The level of the microRNAs can also alter between the normal and the cancer cells, and that alterations can be used as a diagnostic markers. Here the probes for the microRNA are attached onto the surface and then mixed with the isolated microRNA populations of cancer cells and as well as from the normal cells and can be detected. Now the next technique we are going to discuss is the 2D gel electrophoresis. It is a technique for protein profile analysis. First, the isolated proteins from the source, that means cancer cells, as well as a control from the normal counterparts, are first separated based on their isoelectric points. Then they are placed on a second denaturing gel or the SDS page. This technique is actually gives better resolution than the conventional SDS page. Next is the Matrix assisted laser desorption, ionization time of light, mass spectrometry. Mass spectrometry is another technique widely used in proteomic analysis of cancer cells. A mass spectrometer generates multiple ions from the sample under investigation, which then separates them according to their specific mass to charge ratio, and then records the relative abundance of each ion type. As the generation of the sample ion is via the absorption of laser light by the matrix, it is also called matrix assisted laser desorption. The ions are accelerated through the flight tube and the to reach the detector, and then the time taken by each ion to hit the detector varies considerably depending on their mass. This time of flight of each ion is recorded and compared between the normal and the cancer cell sample. Like the nucleic acid based microarrays to detect the transcript levels, protein microarrays can also be designed which uses specific antibodies or other interacting proteins to bind their specific target proteins present in the samples. We can analyze the levels of hundreds of different proteins in a sample in a quick span of time. Tissue microarrays are another way to detect both proteins and RNAs. The probe here is an array of biopsies from multiple cancer samples. They can bind RNAs or proteins from the samples, which can be detected by immunohistochemistry or in situ hybridizations. So today we have discussed the basics about biomarkers and the different techniques which are employed in cancer diagnosis. Hope it is useful and informative. We'll meet again in the next lecture to discuss about different cancer biomarkers. Till then, thank you.